Hi everyone, thanks for being here today. I'll be talking about a part of my master's thesis work that I did here at Bucknell with Dr. Chris Martine, as well as Dr. Angela McDonald, who was the Burpee postdoc at the time, and Dr. Scott Schutte with the Western Pennsylvania Conservancy. And I'll be talking about conservation genomics of the Pennsylvania-threatened plant species Baptisia australis, variety australis. It's an investigation into gene flow in a riparian system. I'll give some background information on this plant that I studied, such as where it grows. Then I'll spend some time on the population genomics portion of my work, and I'll wrap up by talking about the conservation implications. To give some background on the species, it's in the Fabaceae family or the pea and bean family. It reproduces rhizomatously as well as via hermaphroditic flowers that are bee-pollinated. In Pennsylvania, it flowers in late May and early June. It's a perennial, and it's popular in cultivation, so it may look familiar. This map shows the range of Baptisia australis, variety australis. There are two other varieties. However, Baptisia australis, variety australis is the only variety found in Pennsylvania. Within Pennsylvania, Baptisia's distribution is rather limited. It's known from along only 4 waterways shown here: the Allegheny River and 2 of its tributaries, the Clarion River and the Red Bank Creek, as well as the Youghiogheny River further south. Because of this limited distribution and other factors, it's considered threatened by the Pennsylvania Department of Conservation and Natural Resources, and it's considered to be an S2 or imperiled species by the Pennsylvania Natural Heritage Program. It's also considered to be a G3 or globally vulnerable species. Other factors in this conservation rank, aside from the species' limited distribution, include its specialized habitat. It occurs along waterways and open prairie-like systems with prairie species like big bluestem and Indian grass. Habitat loss is a problem for these species, reducing these specialized habitats. This is caused by damming and development, as well as reduction in ice scour, which helps facilitate an open habitat. Climate change, which again reduces this ice scour, as well as changes in precipitation, either resulting in flooding or periods of drought. Lastly, invasive species such as phalaris, shown here, and knotweed both compete directly with Baptisia in its habitat. So now I'm going to dive in and talk about population genomics and the implications for conservation and understanding riparian metapopulation dynamics. That's a mouthful and a lot to wrap your head around, so I'm going to break it down into 2 of my hypotheses. The first being that in Pennsylvania, there will be 2 genetically distinct groups, the Youghiogheny and the Allegheny plus its tributaries. Our second hypothesis is that we'll observe classic metapopulation gene flow between subpopulations of the larger metapopulations. So to give some background on what this means, if you have a metapopulation with smaller subpopulations within it, if all of those small populations were exchanging gene flow equally and all the populations look very genetically similar, you would have a panmitic population. There are other models that have been proposed for riparian populations, such as the stepping stone model, where populations are only sharing gene flow with those right next to them. And then this riparian unidirectional gene flow idea, where gene flow is only occurring from the upstream to the downstream sites with the flow of the water. But what I expect to see is a classic metapopulation with small amounts of gene flow occurring between various subpopulations. Now I'll walk you through my methods from tissue collection all the way to conservation implications of the work. Tissue was collected from 24 sites, resulting in 326 samples being sent out for sequencing. Tissue was collected from 15 plants per site when possible, from 1 meter apart or greater, and various phenotypes were also targeted. And at this point, I'd like to thank Dr. Scott Schutte with the Western Pennsylvania Conservancy for help in the field, but also his knowledge and sharing the ability to access these plants as well as do collections. So now I'm going to walk you through what was done to process these samples once they were collected. DNA was extracted from dried plant tissue, so we put dried leaves into tubes with several different ingredients from a kit and followed some steps and got DNA. It's a lot simpler than what actually happened, but it's all we really need to know for this. We then used something called reduced representation genome sequencing. We didn't actually do this at Bucknell. We sent our samples away to the University of Wisconsin-Madison, and they took the DNA that we sent them and cut it with enzymes. This resulted in lots of tiny comparable pieces of DNA, almost 800 million in our case. So what happens here is we extract genomic DNA, and to compare our individuals, we can't realistically sequence all of those 326 samples, and so we cut out tiny parts of the genome that we can compare across all the samples. Once we got those raw reads back from the University of Wisconsin, we processed it and pulled the pieces and then filtered them. We only wanted pieces that were comparable across lots of samples, and so in the end we only had 350 pieces left across all of our samples, but that's actually a lot for a data set like this. And in the end, we had 318 individuals in our final data set, meaning we only threw out 8 individuals, which is pretty good. That was just because of low coverage of our reads. We analyzed those pieces of DNA and calculated the number of populations and much more with programs in our studio. We also did analyses on Bucknell's HPC cluster BisonNet. Now I'm going to start walking you through some of our results, and I want to point out that I have tagged each of the results slides with what hypothesis they correspond to. So this result, the PCA, corresponds to our hypothesis about genetic groups. And this PCA, or principal components analysis, shows all of our individual samples that we had in our final data set, and they've been color-coded by what population they come from and by what waterway. And these PCs, or principal components, these axes, don't really represent anything concrete. It's just a way to visualize variation between samples and show ordination in space to give you an idea of samples that are more similar. So first up is the Allegheny River here in blue, and I tried to color code from upstream to downstream with light to dark. And you can see that pattern is somewhat followed here in the way the Allegheny River clusters. Next is the Clarion River, which had 2 populations, so there's 2 shades of green, and they cluster together. Then the Red Bank Creek, there are 3 populations there, so 3 shades of red, and they cluster together, but they also cluster rather closely with the Allegheny River. Then there's the Youghiogheny River, and then we have a West Virginia outgroup that I used to kind of compare to see where these Pennsylvania populations lie in relation to other populations outside of the state. And you can see this doesn't give us a concrete answer of how many populations there are in the state or how many genetic groups, but it can help give us an idea. And that takes us to the next analysis, where this analysis is actually designed to give you a number for K, or a number of genetic groups present in your data. This is a DAPC, or a discriminant analysis of principal components. This is very similar to the PCA that I just showed you. It's another ordination plot, but here it's trying to minimize differences between individuals of the same population and maximize differences between those genetic groups that it's finding. And you can see this results in a rather different ordination plot. So although it finds 7 genetic groups, there's a lot more close clustering of some of these. So you have the Allegheny River split into 3 groups: the Upper Allegheny, the Lower Allegheny, and River's Edge, which is below the Red Bank Creek. And you can see that they are kind of mapped in matching colors on this map here on the left. You have West Virginia clustering by itself and the Youghiogheny River clustering by itself, and then the Clarion River and the Red Bank Creek clustering closely with the Allegheny again. This is just another way to visualize those DAPC results. So not an ordination plot, but each of those vertical lines represents one individual. The color of that line represents the likelihood that that individual belongs to the population the program assigned it. So you can see the orange, the Youghiogheny River, and the brown, West Virginia. Those individuals are certainly members of those populations. while the Allegheny River populations and the Red Bank Creek, there's a lot more mixture happening. It's not as clear that those individuals actually belong to those populations. It can be a little confusing, so we'll look at one more analysis. This figure shows something similar to the last plot I showed, where every vertical line represents one sample. It's a structure-like plot created in the program SNMF, and what it shows is a similar pattern to the last figure. It finds 5 genetic clusters. 3 of them are the same, so the West Virginia, the Clarion, and the Youghiogheny. And you also have the Allegheny being split up, similar to before, but it's only split into Upper and Lower Allegheny. And the Lower Allegheny group now contains that River's Edge population and the Red Bank Creek. FST is a measure of gene flow that ranges from 0, being complete gene flow, to 1, being no gene flow shared between 2 populations. This pairwise calculation is shown here in this heat map, with darker colors being higher FST values or less gene flow. It's been determined or generally accepted that an FST value of greater than 0.15 indicates significant differentiation between 2 populations. So here you can see all of the populations that I sampled, and I've grouped them by waterway. So we have the Allegheny River here, the Clarion River, the Red Bank Creek, the Youghiogheny River, and then 2 West Virginia populations that I sampled as a sort of genetic outgroup. I didn't cover that much in this talk, but if you have questions about it at the end, please ask. What this shows essentially is FST values indicate that West Virginia and the Youghiogheny River are significantly differentiated from all other waterways and populations. The same is true to some extent for the Clarion River and the Red Bank Creek. So we're going to bring all these things together now. We're going to talk about what these results mean and recap what we looked at. So the PCA indicates genetic differences between populations on different waterways with an upstream to downstream pattern on the Allegheny River. The DAPC showed 7 genetically distinct populations, and the SNMF plot showed 5. Those FST values on that heat map showed patterns of genetic differentiation between waterways, with limited gene flow between the various waterways. So drawing conclusions from those before we dive into that third hypothesis. The first hypothesis, again, was that Pennsylvania has 2 genetically distinct groups, the Youghiogheny and the Allegheny plus its tributaries. This was not well supported by our various results. And what we decided to conclude was that there were 3 genetic entities that always clustered out separately in all of our analyses. And that's the Clarion River, the Youghiogheny River, and then the Allegheny River plus the Red Bank Creek as one entity. And you might be thinking that there was some finer genetic structure within the Allegheny River and the Red Bank Creek, and that's true, but based on that gene flow and some of our analyses, we can't concretely say that they're all different genetic entities. There's still gene flow happening. So there's some finer genetic structure between the Upper Allegheny, Lower Allegheny, River's Edge population, and the Red Bank Creek, and that needs to be monitored in the future. And our second hypothesis was we would see classic metapopulation gene flow. And what we decided to conclude here was that we see a sort of mixed model. There's a sort of stepping stone model happening based on that FST. The populations that are close to one another have more gene flow than the populations that are further away. We also see some of that unidirectional gene flow, especially on the Allegheny River. And essentially what we're probably seeing is a classic metapopulation with hints of some of that stepping stone and riparian unidirectional gene flow happening. But for right now, we're going to call it a mixed model of gene flow. There are also tangible conservation implications for our work. Because this work was partially funded by the Pennsylvania Department of Conservation and Natural Resources and was in partnership with the Heritage Program and Western Pennsylvania Conservancy, these data can then be handed over to these agencies so they can use them to inform management decisions. It can be used in NatureServe Rank Calculator and the Climate Change Vulnerability Calculator that the Heritage Program uses, and this genetic information can be applied to the idea of management units or element occurrences, This is the number of populations that are managed separately within the state. This is also a unique partnership that allows us to then present this work at the Pennsylvania Rare Plant Forum so all managers have access to this information. I think the main conservation take-home message for this work in Pennsylvania is that there are 3 genetic groups of Baptisia australis, variety australis in Pennsylvania. Previously, the species was not managed as such in Pennsylvania, however, it should be now. And within these groups, there is increasing genetic structure, which is something that we don't necessarily want. So facilitated gene flow might be needed now or in the future, and because we know how the species reproduces, it could be easy to collect seeds and spread them. So having this information could help inform conservation decisions in the near future for this species. And with that, I have lots of folks to acknowledge, including my funding sources here in the upper left, the Western Pennsylvania Conservancy and the Natural Heritage Program, the Martine Lab, folks at Bucknell, and my friends and family. And with that, I'd be happy to take any questions you might have.